this edition of Tissue Talks. It's a real pleasure to have our speaker, Dan Ha, today. Dan is one of those few people that we are inviting back, after they gave a talk some years ago, to hear what is new and exciting in the research and in the field. So Dan comes from the University of Pennsylvania. He had a very interesting career, starting with obtaining a bachelor's degree in mechanical engineering from the Seoul National University in 2000. He went on to obtain the master's degree in biomedical engineering and in mechanical engineering 2 years later. And then he moved to the US, to the University of Michigan, to get his PhD degree in 2007. After obtaining PhD, he joined Don Ingber's group at Harvard Medical School and Harvard University, also at Children's Hospital in Boston, as a postdoctoral research fellow. So from 2009, so this is after his postdoc, through 2012, he was a WIS Technology Development Fellow and also research associate at the WIS Institute for Biologically Inspired Engineering at Harvard University. In 2012, Dan moved back to his native Korea to join the Department of Biomedical Engineering at Seoul National University as an assistant professor. And shortly after, he moved to the University of Pennsylvania, to UPenn, in 2013. He's now a chaired professor of bioengineering at UPenn, and he's also, in parallel to his academic function, he's a chief scientific officer of Vivodyne. This is a company that he founded to foster therapeutic development using organs on chip platforms. Dan is a real pioneer in the organs of chip space. He started his scientific career as a graduate student with a seminal lung on a chip paper that was published in Science, and that really defined the field. So this was really the first time that a function, organ level function of one of our organs was recapitulated in a very simple and highly controllable system. Following this paper that was cited, I think, about 5,000 times, that was really like informing the field, he continued with a series of articles that were published in Science, Nature, Nature Materials, PNAS, Science Translational Medicine. Just last week, he published another seminal paper, this time in Nature Biotechnology, where he is reporting the study of CAR T-cell therapy on a chip. This is like really another major advance. It's very characteristic that designs of Dan's bioengineered models are really based on design principles that are taken from human biology. And let me just mention that, not surprisingly, he received a number of honors and awards. He obtained an innovation award from the Society for Lab Automation. He obtained the Scientific Breakthrough of the Year from the American Thoracic Society. This is for his lung on a chip paper. Best Publication, Best Paper Award, Lush Prize, Design of the Year Award, and Best Product of the Year Award from the London Design Museum. This is like very unusual. He obtained the NIH Director's New Innovation Award, and then one really nice recognition, he is a lifetime member. He obtained a lifetime membership from the MoMA, Museum of Modern Art in New York. Dan, thanks very much for being with us today, and you can now start sharing your screen.
All right, let me put this on first. Okay. So thank you very much, Gordana, for inviting me back. So I checked, earlier today. So last time I gave a talk was back in 2022. So as we were getting out of the pandemic, and so it's great to be back. in 3 years. But so hopefully I will actually deliver something, as meaningful, more meaningful and exciting beyond what I discussed, 3 years ago. And thank you very much for the very generous introduction. Okay, so I usually start my seminar talk with a photo of my entire lab. So I just updated it a couple of days ago. This is my group in action. I have about 13 to 14 people. There's been a lot of turnover this year, actually. Good things happening in the lab, senior postdocs getting faculty positions, and they're leaving the labs. And so we're equally split between postdocs and students. And I would say, my lab is very, driven by questions and problems. So we're driven, the question-driven, problem-driven lab, I would say that. But we work on many different projects. Our interests are all over the place, but one common thread that runs through these different projects actually that we work on is that we, use this, bioengineering technology called organs on a chip, and this is actually kind of, I don't think I need to explain the concept of this, technology, to this audience, but so this is actually, when it comes to technology and tools, we tend to identify ourselves as a group actually at the leading, at the forefront of this field of organs on chips. So that's actually the main technology we've been using to address, these interesting, very challenging questions and problems that we encounter in medicine. So just to get everyone on the same page, just to briefly explain what we do in the lab. So our work basically revolves around this idea of creating microfabricated devices for cell culture. But very importantly, when we grow these cells, human cells in these small-scale devices, we use microengineering technologies to provide the cells with the type of environment that they would like. They would feel at home, if you will, so that they do what they're supposed to do. So we try to cells, into thinking that they're sitting in the body so that they can build themselves into complex tissues. They regain their, functional capacities. And so by using these devices, our goal is to engineer realistic, living human tissues that can be used for, biomedical and environmental applications. So that's kind of the general, principle that we follow in our work. So these systems are generally called organs on chips, but as this field actually, we've seen a very, very, significant growth in the field. And so the organ chips, this term is now used synonymously with tissue chips or, microphysiological systems, as many of you know. And another term that is gaining increasing, popularity in recent years, especially, is this, kind of umbrella term, complex in vitro models. Okay, and as many of you know, these complex in vitro models are playing a really central role in this new, exciting, I would say, global movement to develop and, adopt what's known as a new approach methodologies or new alternative methodologies, in short, NAMs, which has been highlighted many times in recent years, especially in FDA's, Modernization Act, and also, FDA made a groundbreaking announcement this year, back in April, to start, phasing out animal testing for certain classes of drugs. So these are very exciting times, for us as model developers. So using this organ chip, MPS approach, I would say over the last 15 years or so, my lab has developed a variety of MPS, human tissue model systems, some of which are shown here. So my goal today, given the fact that I actually spoke about some of these model systems 3 years ago, my goal is to really focus and highlight recent advances in the lab. And I'd like to actually organize this discussion around these three questions, I've been thinking a lot about over the last 5 years. My lab has been thinking a lot about over the last 5 years. So this is the title of my talk, the three questions that we've been using to guide our ongoing and, emerging efforts to really advance organ chip technology and complex in vitro models in general. And these three questions, I would say, I'm going to cover three pillars. So three focusing areas that I would like to, cover today are biology, data, and translation. So these are three important aspects of complex in vitro models or MPS technology I would like to, talk about today. So let me actually get to the first question. So the first question is, shown here. So can we actually make these complex in vitro models, organ chip models, more realistic? So that's actually one of the questions, my lab has been asking a lot over the last 5 years. I always actually tell my trainees that, your models don't have to be, perfect, but, you want to make these model systems useful. But in order to make these model systems useful, what matters at the end of the day is the specific question or problem you're asking, you're trying to address. So the general principle that we follow is that the complexity or realism of the in vitro model you're developing is determined by the specific question you're asking. But one very important trend that we see in medicine is that these questions we are asking, the problems we're trying to address, they become more and more complex, complicated, and also multifaceted. So I definitely see the need for more sophisticated, more realistic, more biologically complex model systems that we can use to address those more challenging questions. So how do we actually make these model systems more realistic? So that's the first question. So, to talk about this question, I'd like to spend just a 1 minute actually talking about kind of the general design principle that we as organ chip engineers use to build these organ chip systems. How do we actually make these organ chips? How do we go about designing and fabricating organ chip systems for research in biomedicine? So this organ on a chip, term could be misleading in that obviously it's not the whole organ represented by, this micro device, these devices that we make. What we do is essentially when we have a target organ, the first, step is to identify the functional elements, functional units of your target organ. And once you do that, then you go in and take a closer look at those functional units to understand what type of cells there are, how they are organized, in three-dimensional space, and very importantly, it's actually we also need to understand what kinds of biochemical, biophysical, environmental cues they experience in vivo. So once you figure these out, then the last step is to go ahead and design a chip that will allow you to replicate those some of those essential features of your target organ, especially the functional elements of your target organ. So let me give you an example. So let's say you're working in a drug company and you're tasked to develop some kind of a complex in vitro platform model system for the purposes of testing ophthalmic drug the company is actually developing, right? So then in that case, your target organ would be the eye, but eye is a very complex organ. So in this case, the specific kind of the anatomical element or unit of the eye that you want to model would be the ocular surface, front window of the eye. So consisting of the cornea, and then also the conjunctiva, as you know. And we know that these tissues are sitting in a very interesting environment. So first of all, there's this thin layer of tear fluid formed by blinking activities, and so this layer acts to hydrate the surface. It forms a smooth refractive surface for light transmission and so on. So to model this functional unit of the eye, we could actually design a device like this. So this is actually was one of the first projects, in my lab. And so what we did was we actually used these highly porous three-dimensional scaffolds that looked like contact lens devices, as shown here. And we came up with the way to deposit and grow these two different cell types, human primary corneal cells at the center, the convex, surface of the scaffold, and we were able to surround them with, conjunctival epithelial cells. So this is actually how we engineer the tissues. And very importantly, as I said earlier, it's actually very important to expose these cells to the environment or put these cells in an environment that is very similar to what they experience in the body. So to achieve that, we actually expose this confluent layer of cells to air to induce differentiation. So as a result of differentiation, the cell For example, corneal epithelial cells that start off as a single layer become stratified, forming, 5 to 7 layers. They form very tight junctions. They upregulate their differentiation markers. And initially, their surfaces are flat, but they actually form these beautiful microstructures that are very similar to, what's shown on the left, right here, actually, taken from the surface of the human eye. And once you engineer these tissues, then what you can do to mimic that environment, dynamic blinking environment, is to use a very soft elastomer as a biomimetic eyelid. And then you can actually use the electric motor to slide it back and forth over the surface of this scaffold to form actually tear film. And so this is actually how we engineered the system, and we were able to use the system in this collaborative study with a pharmaceutical company that was developing ophthalmic lubricant actually, at the time. So we tested their drug in this system to show its friction lowering effects, and also we discover unexpected anti-inflammatory capacity of this drug thanks to this model system. Okay, so I would say this is actually a good example of organ on a chip technology, and where useful organ chip MPS system. But I'm actually presenting this as an example of more of a conventional, approaches to creating complex in vitro models. So if you remember the question I was asking earlier, how do we actually make these model systems biologically more realistic and complex? So as we, began to think more and more about these questions, we became really interested in this idea of using tissue and organ development during embryogenesis as a source of inspiration. So what I'm showing here in this movie is a very complex respiratory system being developed in the Drosophila embryo. And it's amazing, actually, how these stem cells and progenitor cells, they're instructed by biological signals coming from the surrounding environment. They somehow know where they go, and they undergo this very precisely controlled differentiation, fate specification, and they also undergo very strong dynamic and dramatic structural reorganization, and they self-organize into these very complex tissues. So it'll be quite challenging, if not impossible, to mimic really the inner workings of these processes. But our question was simple: could we actually try to mimic certain aspects of these developmental processes and use that as a kind of a new principle to building a new approach to building more biologically more complex and physiologically more realistic tissue systems in our experiments. So the first example I would like to show you actually today is bone marrow on a chip. More specifically, we published this paper last year talking about how to actually how we can use these microengineered 3D culture systems to mimic the hematopoietic vascular niche in the human bone marrow. So briefly, the bone marrow is actually one of the organs that developed the last, right before delivery, and so these hematopoietic stem cells actually they arise from hemogenic sites in different parts of embryos, especially from blood vessels, endothelial layer, and then they move around a lot through the embryo, receiving signals needed for expansion and maturation. But eventually, they're known to kind of relocate and get into this medullary cavity, hollow space at the center of the bone, and then they kind of populate this area. But it's known that this process of HSC colonization is known to occur actually concurrently with the development of these blood vessels, sinusoidal blood vessels. So kind of learning about these developmental processes, our question was, starting up was very simple. Could we actually mimic this concurrent process of HSC colonization and vasculogenesis in the medullary cavity in the bone? So one of the first things we had to figure out was to engineer realistic human blood vessels. So to do that, long story short, this is actually one of the devices that we've used to achieve this goal. And this is a cutaway view of this device. And what I'd like to highlight is that there are three partially separated compartments defined by these two protruding microfabricated structures. And so using these microfabricated rails or pin structures, you can actually trap liquid only in the middle compartment. So this is actually a pretty well-known physics phenomenon called capillary pinning effect. And so you can inject gently a mixture of liquid and cells into the device like this. So using this, method, we were able to form this hydrogel embedding, and then we were able to embed fibroblasts and endothelial cells in this hydrogel. So after gelation, what you can do is to fill the side channels with media to feed the cells in this 3D culture system. And we also seed side channels with endothelial cells. So during this 3D co-culture, what happens is when the environment is actually, the right environment, quote unquote, right environment, the endothelial cells, if you think about it, what they do in the body is to form blood vessels. That's exactly what happens in this ECM scaffold. They assemble themselves into 3D vessels, and they also anastomose with the endothelial lining, monolayer, in the side channels. And that's how these blood vessels in 3D scaffold become accessible and perfusible, between these two side channels. So let me actually show you an actual movie of this, vasculogenic process happening in the device. So this device actually, you're looking at from a top, and this is a bird's eye view of the device. It's a fibrin scaffold containing RFP expressing endothelial cells. So as you can tell, over time, they become elongated. These cells become elongated. They connect with one another to form a network structure in the scaffold. So this takes about it depends on the type of endothelial cells, but takes somewhere between 5 and 7 days. And as a result, we can form these really, pretty, realistic blood vessels in the scaffold that are very similar to human microvessels, as is shown here. And what's really nice about this system is that using these two side channels on either side, we can actually perfuse the entire network in a semi-controllable manner. So as is shown here. And so this is actually how we are able to engineer realistic human blood vessels. But in the marrow, what happens is, vasculogenesis happens concurrently with the colonization of that environment by HSCs, hematopoietic stem and progenitor cells. So to model that, we actually use exactly the same device, same design, but we added CD34 positive human hematopoietic stem and progenitor cells. And Andrei Georgescu, fantastic grad student who was working on this project at the time, spent somewhere between 6 and 8 months trying to figure out and optimize co-culture conditions actually. So we had 3 or 4 different cell types, but when the conditions are met, these cells kind of self-organize into this very complex tissue shown here. So what's stained in green are blood vessels, and these red cells are blood cells, form in this marrow on a chip system. So we actually went in and really looked at what type of very heterogeneous cell populations form in this device. We saw a wide variety of actually blood cells in this environment. And these are some of the images of erythrocytes, red blood cells formed in this device. We even saw erythroblastic islands actually consisting of the central nurse macrophages surrounded by erythroid cells. And depending on media conditions, we were also able to form very mature myeloid cells, granulocytes, and mature neutrophils, as is shown here. So, but very importantly, we were also able to show, in this study that this hematopoietic vascular niche engineered in this device actually allowed us to maintain stem and progenitor population for prolonged periods, as verified by CFU assay shown here. So when these actually blood cells are formed, so let's say these are granulocytes actually formed within the hematopoietic vascular niche in this device, they kind of hang around, near the vessels. But when they're stimulated, using drugs or pro-inflammatory cytokines, we observe some of these cells intravasate and then flow out of the device actually, because the vessels were perfusible. So In one study, what we did was we created bone marrow on a chip, and then we connected to an airway on a chip, shown here. So the airway on a chip had three compartments separated by two intervening membranes. So in the top chamber, we cultured small area epithelial cells, and then we fully differentiated them under air. And the middle compartment was designed to mimic the interstitium, basically connective tissue containing fibroblast, and the bottom compartment was lined with vascular endothelial cells. So what we did was we actually infected this airway tissue. Our goal was to, see if we can mimic innate immune response to lung infection, driven by the marrow. So we infected the airway tissue with Pseudomonas, and as you can tell, a lot of interesting things happened, including, the Pseudomonas actually cells, kind of compromise the epithelial barrier. But very importantly, the infected airway tissue secreted a lot of chemokines and cytokines. And because those two devices were fluidically connected, these secretory factors flowed into the marrow chip. And what we noticed was, when the infected airway chip was connected, the number of cells coming out of the marrow chip, increased dramatically. And a vast majority of these cells were CD66B positive mature neutrophils. And these cells flowed into the area on a chip. And what we also noticed was number of cells going in was much greater than number of cells coming out. So we knew something was going on in this area on a chip device. So we used the scanning electron microscopy to visualize interesting events happening within the area on a chip. So as you can tell on the vascular side, these neutrophils adhere to the endothelium, they undergo transmigration, and then they pop up on the epithelial surface and they move in a directional manner towards bacteria, and they phagocytose, and we even saw netosis, and this is a citrunyl CYTH3, and we even saw a neutrophil swarming. So if you think about it, there's nothing new scientifically here, but being able to, kind of emulate the entire process, entire cascade of innate immune response to, airway infection was, something, that really, it was a very exciting, actually, demonstration, exciting accomplishment, that we showed in this, study. So in collaboration with GSK, we actually used this system to test myelotoxicity of anti-cancer drugs. In the interest of time, I'm going to skip, over this, but we also succeeded in scaling up, this platform, and we were able to generate very reproducible data indicating the cytotoxic potential of anti-cancer drugs on living cells in the marrow. Okay, so let me actually check the time here. Okay. So again, just to get everyone on the same page, I'm talking about kind of, this question: can we make organ chip systems, complex in vitro model systems, more realistic? So I don't think, this discussion would be complete without talking about organoids. And so about, 6 years ago, I was approached by a senior editor in Science, actually, and he actually asked me if we could, write a perspective article, review article, to talk about how organ chip technology can help advance the field of organoids. So, we actually talked about how these microengineered complex in vitro, model systems and platforms can build, more enabling, systems for organoid cultures, and how we can achieve more realistic and more predictive in vitro models, combining both organ chip, platforms and stem cell derived organoids. So this was actually a very interesting kind of opportunity, very great opportunity to think about kind of the what we might be able to achieve at this intersection between organs and chips and organoids. So over the last, 5 years, actually, my lab kind of really tried hard, actually, worked hard to demonstrate kind of the power of microengineered cell culture platforms for organoid culture in general. And so this is actually one of the examples we published, last year, or actually a couple of years ago, about this, platform called Octopus. So this is a platform basically designed to be tissue culture inserts And you can actually use the system to eliminate or minimize diffusion limitations, commonly present in, drop culture, systems people use for example, intestinal organoids. And so using this system, we were able to show that those organoids didn't have to be passaged for over a month actually, and as a result we were able to grow these organoids much bigger. And it's not just the size, we were also able to really push their maturity as verified by single cell sequencing data. And we were able to do similar things using patient-derived organoids. And in this case, we actually obtained cells, intestinal stem cells from IBD patients. We were able to form more physiologically relevant or realistic IBD organoids that really express disease phenotypes, much more readily. And then in the last figure of this paper, we're introducing this platform where we can actually vascularize and perfuse intestinal organoids, both healthy and diseased intestinal organoids, to model that parenchymal vascular interface. And this is actually an interesting, really interesting study. And so this is actually a new study we haven't published yet, but another major type of organoids that we've been working with in my lab are lung organoids. So we've been working with both alveolar organoids and airway organoids. And so this work was partially driven by this actually interesting observation. So what I'm showing here is an MRI movie of a developing fetus, and I'm not sure if you can, see this clearly, but so this is where the fetal lungs are located, and so it turns out that fetal lungs actually move a lot. So the fetus, sometimes swallow amniotic fluid during which the lungs can expand and contract. And lungs happen to be anatomical sites, where a lot of fluid is generated. So when the fluid actually exits the lung, lungs can also undergo, very interesting and dynamic mechanical movements. So in general, these movements are called fetal breathing movements. And it's known that fetal breathing movements, frequency and amplitude of these movements, increase actually at a later gestational ages. But interestingly enough, actually, these later gestational ages happen to be the period of pregnancy where the lung undergoes very dynamic and a dramatic branching morphogenesis to form these alveolar air sacs, critical anatomical functional units of the lung. So, connecting these two dots, we kind of thought to ourselves, or we hypothesized that perhaps, these, more frequent breathing movements, more prominent breathing movements, and especially the forces generated by these breathing movements might play a very important role in this lung, development, morphogenesis, and also tissue maturation, alveolarization in general, which is critical aspect of lung development. So to kind of test and study, these interesting questions, actually, so we build a micro-engineered 3D organoid culture platform. So this work was initiated and developed by Estelle Park, who is now a BME professor at Purdue, taken over by senior research associate, Dr. Min-Suk Kim, in my lab. And this was done in collaboration with Daryl Cotton. He's a big name in lung stem cell biology and organoids. So let me just show you a snapshot of this, project. So this is actually what we saw. So using our device, we were able to grow these organoids in a physiologically relevant conditions, actually, that include mechanical stimulation, mechanical, deformation of these developing organoids. And as a result, we were able to really, induce and increase the extent of branching morphogenesis and also enlargement of these organoids, as you can tell. It's not just the shape and also structure. We also saw significantly enhanced tissue maturity, maturity of both alveolar type 1 and alveolar type 2 epithelial cells. So, and we conducted very extensive sequencing studies to really understand the underlying mechanisms of these really interesting mechanobiological, phenomenon. So this actually study is being concluded, and we are hoping to submit this manuscript soon. But this is another actually project where we try to build more realistic models to address interesting questions in biology and medicine. Okay, so let me go back to the this question and summarize this, first question. So can we make models that are more realistic? I think this is a very has been a very important question for the lab, and so I think the takeaway from this discussion is I always actually emphasize to my trainees that having simple yet useful models is always the best. Okay, but in situations where you need to build complex model systems, uh, definitely, this idea of using development, okay, development of biology as a source of inspiration, I think it's a very exciting and, promising approach as we've demonstrated in those couple of new papers. Okay, so that is a question about biology. So let me get to the second question. So second question is about data. So the question we've been asking ourselves is, can we make models, that are more informative, that can produce, useful data? So I think that as a field, there's been a lot of focus on creating, more realistic, more sophisticated, model systems. But if you think about it, it's really the data that matter at the end of the day. So what kinds of data can we generate? And more importantly, are they useful for anything, right? So this is really was a data-focused, data-centric investigation we've conducted over the last 5 years or so. So this actually, the example I like to focus on, in the context of this question is this new paper that Gordana mentioned during her introduction. So this is a new paper that just came out in Nature Biotech talking about tumor on a chip that we've developed to model CAR T immunotherapy. So using the same platform and 3D culture techniques I talked about earlier, what we can do is we can include tumor spheroids or small pieces of tumor explants in the same hydrogel with blood vessel cells and fibroblasts to form these heavily vascularized tumor constructs. So Haijiao Liu actually is a lead author of this paper, actually came up with this really cool idea of creating open top, device where he would actually form this open top chamber and then initially block it with this insert. And then he would form blood vessels first and make them perfusible, get them ready. And when these tumor constructs are available, they become available, then we get the tumors from patients or CDX models. We chop them into small pieces and then open this insert or the chamber and then drop them into these wells. And after which these tumors that had been transplanted into this in vitro device could be vascularized and perfused. So using this method, actually, we can engineer 3D tumor constructs with blood vessels surrounded by fibroblasts, and these tumor constructs can be perfused using these two-side chambers. So what we did was we actually used this platform to study CAR T therapy of solid tumors. And in the interest of time, I'm not going to go into details of the CAR T therapy or what clinical challenges are. But as many of you know, CAR T therapy, there's been tremendous success in using CAR T therapy for blood cancers. But now a lot of efforts are being directed towards using this approach to treat solid tumors. But um, it's not easy to make this happen, and there's been very limited success in this therapeutic approach for many reasons. But one of the most important reasons is that these solid tumors are surrounded by very complex ecosystem tumor microenvironment. It's not very easy to get uh, engineered immune cells, CAR T cells, into the tumor microenvironment, tumor tissues, and then also make them kind of exert cytotoxic functions for prolonged periods. So what we wanted to do was to major knowledge gap in the field is actually how are these CAR T cells interacting with cancer cells in the tumor microenvironment? So to really kind of contribute to these kinds of studies, we wanted to actually kind of demonstrate the capabilities of our model to emulate these three essential steps of a CAR T cell trafficking and function. So basically, CAR T cells circulating in the bloodstream would have to recognize the signals from the tumor microenvironment, then they would have to get out of the vessels and then move towards the tumors. And then once inside the tumors, they would have to exert cytotoxic, antigen, directed cytotoxic effects on the tumor cells, and they would have to have a persistence, actually persistent function there. So can we actually model these important kind of steps or processes that determine the success of CAR T therapy? So let me show you just a snapshot of this. You can read about this, work in the paper. But so this is one example where we vascularized CDX tumors engineered to overexpress this tumor antigen called mesothelin. So we call these mesotumors. And we perfuse these tumor constructs with CAR T cells engineered to target mesothelin expressing cells. As you can tell, this is, the minutes after CAR T cell infusion, many of these cells actually stick to the tumor-associated vessels. They look very activated. They crawl around. And using this actually transparent device, we can actually see through kind of the layers of the device to see, observe what happens. And so as you can tell, these actually CAR T cells come out of the extravasate, and then they move towards tumors. And we can track them over time to see, to really, to monitor the progression of CAR T cell trafficking. And we can also follow, these tumors over time to really, to quantify changes in tumor volume and tumor area. So in this experiment, we generated mesotumors that are overexpressing mesothelin and control tumors that expressed very low levels of mesothelin. As you can tell, there is a significant difference in terms of CAR T cell trafficking and also tumor growth over time. So, but our goal was not create a realistic tumor model. So our goal was to, really investigate, whether having this realistic model would offer any benefits in terms of data production or generating knowledge and insights. So one of the first questions was, what kinds of data can we really get from this, realistic in vitro model of CAR T therapy? So one of the first things we did was because we were able to extract tumors and then only isolate, harvest T cells that have infiltrated into the tumors, and you can differentiate them from these CAR T cells in the surrounding stroma. We actually flow them, and we kind of compare their phenotypes in terms of marker expression. So as you can tell from here, I actually don't have time to go into all the details of these data, but the tumors, the CAR T cells inside the tumors, they exhibited, effector memory phenotype and also tissue resident, T cell phenotype and so on. And we were also harvest these tissues as a whole for single cell sequencing analysis. And this analysis gave us a lot more data in terms of, how these CAR T cells are changing their phenotype as they engage their targets, and also how these tumor cells were responding to these CAR T cells in both, control and mesotumor groups. And using, cell phone DB and similar tools, actually, we can conduct ligand receptor, interaction analysis, as many of you know, and this really allowed us to really, dissect and, probe cell-cell interactions happening in our model systems. So, through these, different analyses, we were able to kind of demonstrate the possibility of generating very, high dimensional, very informative data from engineered, microphysiological systems. But this was a very interesting result. So when we looked at the CAR T cell endothelial interactions, so as you can see here, so this is a CAR T cell side, and this is the endothelial side. So CXCL10, CXCL11, these are chemokines, so ligands, that bind to CXCR3 receptor. And this actually is known to induce T cell recruitment. It's a very well-known signaling axis. But what we found, actually, what we discovered in our analysis was that, these very well-known pathways were being interfered by this enzyme called DPP4, produced by, some of these cells, as shown here. So our question was very simple. Could we actually inhibit the activity of this enzyme? And as a result, could we actually increase CAR T cell trafficking into the tumors? So fortunately, Haijiao actually found this FDA-approved drug, vitagliptin, that is currently used to treat type 2 diabetes, and it's known to also inhibit DPP4 activity. So when we treated the tumors with CAR T cells and vitagliptin at low dose, we didn't see much difference in terms of tumor growth, and also CAR T cell trafficking slight increase. But when we treat the tumors with CAR T cells in conjunction with high doses of beta-glucan, we saw arrested tumor growth as well as a significant increase in CAR T cell trafficking. So, and we were able to quantify the tumor growth over a period of 20 or more days. So as you can tell, the data actually generated by this MPS system, made it possible for us to discover this kind of new, signaling activities, which could be pharmacologically modulated to increase CAR T cell trafficking. And in collaboration with Josh Jovanovich's lab at Princeton, we also conducted unbiased metabolomics analysis to identify potential biomarkers that could indicate this increased efficacy of a combinatorial therapeutic approach that includes both CAR T cells and beta-glucan administered at high doses. Again, you can read more about this study. There's so much data. It took us like 2 years to publish this paper. So again, I hope you enjoy this paper. So I don't have much time left, but basically this is a new paper that's coming out in Nature BME where we talk about asthma on a chip. So asthmatic tissues, we're actually showing that somehow they're very, very sensitive to mechanical forces. During asthma attack, they're subjected to very, dynamic compressive forces. Normal cells don't really respond to those forces, but asthmatic cells are very sensitive. And as a result, they actually make tissues very fibrotic, and also blood vessels actually, remodel, and vascular density increases significantly. So in this paper again, it's kind of along the lines of using these MPS system as data generators. We actually analyze proteins inside the cells and secreted proteins to identify really the key, molecular mediators of these disease actually specific, tissue remodeling. And we were able to identify some potential therapeutic targets and even tested, a couple of candidate molecules actually to show their efficacy. And this is actually another paper also accepted in Nature BME, coming out in Nature BME, a fantastic study done by Puriya Fatai, who is now a professor at UGA, University of Georgia. So he actually created this very realistic model of maternal-fetal interface, and he used the system to mimic cadmium exposure during pregnancy. And using this, we were able to really show, potentially detrimental effects, adverse effects of cadmium, in the maternal circulation on the fetal tissue. And more importantly, with Josh Jovanovich's lab, we identified potential biomarkers that could indicate biological adverse effects of cadmium on both fetal and maternal tissues. Okay, so just to quickly conclude this, question, this theme, so I think this is something we as a field actually should, make more efforts towards it. So I think creating, realistic, sophisticated model systems is a lot of fun and very important, but, I think the bar is getting higher, and if you will, and so we want to actually, be able to show, these are not simply just biologically sophisticated and technologically more sophisticated model systems. They have great potential as data generators, as data engines that can produce, very useful information that could inform, clinical developments of new therapies and, biomarkers and so on. Okay, so I only have, I'm supposed to stop now, but, very quickly. So my last question, I haven't gotten to the last question, but so let me actually share with you this interesting story, okay, very quickly. So let me go back to this Iona chip, and so this was a huge success as a research project, and we got we had this Nature Medicine paper, and a lot of press coverage and so on. So after this paper was published, we were approached by one of the major pharmaceutical companies out there. I'm not gonna, reveal their names here, but so what they wanted to do was to use the system, fantastic system that they saw in the Nature Medicine paper to see if they can recapitulate this interesting corneal pathologies, they saw in those patients receiving, anti-cancer drugs, being developed at the time, actually. So we're glad, and actually we gladly took on this opportunity, and we conducted a sponsor research agreement, And so this is actually what we initially showed in the paper. But unfortunately, the fantastic student who developed this project, who was leading this project, left, and this industry engagement happened well after she left. And so we had to hire new people to reproduce some of these results and get this system back up and running. Despite months of effort, this is actually what we got. Like, the cells actually we were using were discontinued. Actually, we had to seek additional alternative cell source, and then we also had to change media. Despite many months of effort, this was the best we could get actually. So this tissue made everyone happy, but we were baffled and we were actually really frustrated by this results. And so this actually, a long story short, this actually became a very inconvenient truth actually to me and to my lab. Despite the fact that we had such great success as as a research project and publication, but we try to actually make this platform useful for potential end users, drug companies, and we failed miserably. So this is actually a very, very important question. My lab has been thinking a lot about over the last, 5 years. How do we actually make this system more useful, practically useful? Okay. So there are actually, many issues but, I just talked about poor reproducibility, and it's so much work to make these systems. And so it's really becomes really difficult to make these experiments, highly reproducible. And we try to actually move these systems from one lab to another. Oftentimes we fail. And another major issue is actually scalability. So because so much work to make these model systems, it becomes a huge challenge to run many parallel simultaneous experiments to obtain sufficient amounts of data that we need to understand, complex biology or to make accurate preclinical predictions and assessments. So this is actually the last question. I'm going to actually end this, presentation with a couple more slides. How do we actually make these models? I don't want to make any provocative statement here, but again, please keep in mind that these are the questions, specific to my lab. But in general, how do we actually make these model systems more robust, more translatable? Okay, so that's a really important question. So as part of this effort, let me just very quickly show you. So one of the most exciting advances in the lab has been to develop this, higher throughput system. So this is actually a 6-inch wafer device containing over 200 individually addressable culture chambers, designed by Andrei Georgescu, who's a phenomenal engineer. And he actually came up with this kind of idea analogous to how electric circuits work. And he designed this pneumatically driven fluidic valves and pumps actually to individually control, what goes into each of these chambers, and you can program fluidic actuation and culture and so on. You don't have to use these sophisticated valves and pumps. You can just design these devices to be open top and you open wells. And this is actually one of such examples of such devices. And it's a high density array containing 256, individually addressable cell culture chambers. And to really address the reproducibility issue, we strongly think that belief that it's human actually that causes all these reproducibility issues and errors. And so we have championed this idea of using automation as a strategy to overcome the issues of poor reproducibility. So we build and use robots to eliminate humans, from the workflow entirely. So, and as a result, what's really exciting is not technological advances in terms of automation or manufacturing, but it's really the opportunity to increase the amount of data, okay, amount of high quality, high dimensional data that we can get from these supposedly better, much more advanced human relevant in vitro model systems. I think this is a truly unprecedented opportunity, and we have an opportunity to really transform the paradigm of how we model biology in biomedical research. And so based on this vision and technical advances, actually Andrei Georgescu, my former student, and myself actually started a company called Vividyne. Again, we offer very realistic tissues, fully automated, and also we can generate ton of data compatible with AI scale analysis. And so this is not a company selling these, chips or instruments. So this is a kind of the data, data generating platform, data engine, I would like to call it. And we use actually robotics to fully automate the process. This is a slightly outdated, version of this movie, but as you can tell, we can actually automate the entire process, from cell culture, seeding, tissue engineering, analysis, and using robotics. And as a result, we can, really run tens of thousands of experiments at the same time to really, drastically increase the amount of data. And these data are high quality data, high dimensional data generated by human tissues. And so this opportunity to generate human data before clinical trials is what's driving us. So with that, sorry for going over time. I'd like to thank my group, my collaborators, and my funding sources, and I'll be happy to answer any questions. Thank you very much.
Thank you so much, Dan. I mean, thank you for the great science. It's really mind-blowing. Thank you for the philosophical messages about what is important. And also thank you for this last part when you very honestly discuss some of the issues that we are all aware of, that actually things work differently, in the research space and translational space. And I think these are some of the issues that we should be really thinking about. So two things. Let me just announce the next speaker. So this will be next Wednesday. We have Kelly Stevens from the University of Washington in Seattle, who will be speaking about bioprinting human liver. So we have very little time left, but I hope then that you will agree to answer these questions directly. We will give you contact information for people, and maybe we can take at least like one or two questions during the time that is left. The
Sure.
first question is from Prasida Guragain. Is our flow and shear an absolute necessity to form endothelial networks from endothelial cells? Were the endothelial networks stable throughout your experiment in the bone marrow model?
So great question. It's been our experience that shear stress is not an absolute necessity actually to form endothelial or blood vessels in the device. So we're still in the process of figuring this out. So what happens during vasculogenesis and anastomosis of these 3D vessels with the endothelial lining the side channels is that as they connect with one another, so we typically don't use flow pumps to maintain these devices. We typically use enlarged reservoirs to introduce a sufficient amounts of media, and we change media more frequently every other day, every 3 days. That's how we maintain these tissues. But as these blood vessels form and they connect with the side channels, as soon as they connect with the side channels, we saw the flow of media from the side channels into the gel, through the gel to the other side, and so on. So there is a little bit of a fluid movement that happens, in these tissues, but over longer periods of time, we don't actually induce active, flow through these tissues. So but as you know, there's ample evidence out there in the literature, and also, for example, Chris Chan's lab has done, extensive amount of work to show the importance of shear stress on endothelial phenotype and function. So I think it really depends. So the downside of actually inducing flow through the vasculature is actually it's not very straightforward to come up with a way to perfuse like tens of replicates, hundreds of replicates at the same time. So this becomes an interesting engineering challenge and instrumentation challenge. And so and there are, as you know, probably alternative methods, like you could actually use a static configuration, but you could place your devices on a rocker actually to induce back and forth flow. That's been used by many labs in the field. But going back to what I was just saying, it would depend on again the question you're asking. So if your question is really centered on how endothelial tissues respond to certain factors, or if you're studying endothelial, phenotype or changes in endothelial structure and function as a result of disease, for example, then I think it's you would have a reason, to maybe think about creating a flow setup to shear the endothelial cells at physiological levels of shear stress. And so, but in general, from the perspective of building these models for, and conducting experiments at reasonable throughput, we usually don't flow, media through the vasculature.
Yeah. Thank you, Dan. Thank you so much for this really inspirational lecture. And all of you who ask questions will get your answers soon directly from Dan Ha. Thanks, everyone, and see you next Wednesday.
Thank you very much.
Sure.